when you think of a kinase enzyme or a kinase receptor, basically within a cell there's things that are inactive, right? And a kinase basically activates them.
Okay.
And it's kind of like a cascading effect. So there's this picture in the board that I really like a lot. Let me pull this up real quick. So let's see how to explain this. So, all right. Well, with this particular one, we've got like this receptor, right? And let's say there's all these inactivated kinase proteins, right? So, or not kinase, but proteins within the cell, right? So what happens with this type of protein is, let's say a hormone binds to the protein. What that causes is this protein to become phosphorylated. So it gets all these phosphate groups, right? And then because it becomes phosphorylated, it's able to phosphorylate, or wait, let me double check here. Yeah, it's able to phosphorylate all these proteins, right? And then when these proteins are phosphorylated, they're activated and able to initiate a cellular response. So what's cool about this is that from one hormone binding, we've got multiple cellular responses, right? Because each one of these are going to start a cellular response, right? And so that helps kind of, it's kind of like a snowball or a cascading effect, right? Okay. So we don't need one hormone for every cell response. We can have one hormone and have multiple cellular responses. Okay. And that's why, levels of hormones, you've probably talked about all these feedback things and stuff. It's really important that these hormones are at the proper level, because if they're too low or too high, you can either have way too much of a cellular response, because for each extra hormone we're creating way more responses, right? But also for each one we're taking out a bunch of them. So that's why it's so important for it to be balanced. But there's several ways that this can happen. So this is like the membrane bound receptor becomes phosphorylated, but there's also like the G protein receptor you've probably heard of, which has got like those 3 subunits.
Alpha, gamma, beta?
So it's usually gamma, beta, alpha, because the alpha one is the one that comes off.
Gotcha, that's right.
Activates an enzyme, whether it's adenylate cyclase, guanate cyclase, or phospholipase C, I think, and which just depends on whether it uses ATP, GTP, right? If it uses ATP, it's adenylate. If it's uses GTP, it's guanylate. But basically what this does, it converts this into cyclic AMP or cyclic GMP, right?
Yeah, what does cyclic AMP do?
So the cyclic AMP and cyclic GMP are known as second messengers, okay? So you can think of like the hormone binding is the primary messenger or the first messenger, right? And then it initiates a response and starts a cyclic GMP and AMP, which are the second messengers, which then those go on to do the similar thing like this, activate protein kinase, right? So these are able to activate these protein kinases and initiate cellular response, right? So it's another way of kind of getting a similar response, but different hormones bind to different types of receptors. Like maybe this one is more fast acting, right? But you end up not being able to phosphorylate as much, right? Because with this way, with this we're making a ton of cyclic AMP, cyclic GMP, so we're activating a ton of. So this picture is pretty good. It kind of shows how this is going to work. So you've got your hormone binder receptor, activates G proteins, activates the enzyme, which makes all these, which activate all of these, right? But with the phosphorylation one, it can only phosphorylate so many proteins within the cell, right? So that's kind of how that works. Sometimes there's a different one where, so this enzyme right here is if it uses so this is like ATP, adenosine triphosphate, right? So this is adenosine monophosphate, but it has a different structure, so it's cyclic AMP, right? So if it uses ATP, this is adenolate cyclase, right? Or I want to say, is it guanate or guanolate? Let's see. Adenolate cyclase. I'm going to give you the correct name here. Guanolate.
Yeah, they use that. There's a question that has that in it, I didn't know.
Yeah, and both of them are guanolate cyclase. Okay, and then, so if it uses ATP, it's adenolate cyclase. If it uses GTP, it's guanolate cyclase. Then there's also another one called phospholipase. Let's say C, or is it C? Phospholipase C. Basically, what that does is it converts so this one, in this process right here, takes IP2, converts it into IP3, okay? And this always activates calcium channels within the cell, and that calcium binds to other things, which causes a cell response, right? So the end result is always some type of cellular response, but depending on what type of hormone it is, determines like what kind of enzyme it uses, and which kind of determines which, because cyclic AMP might activate different protein kinases than GMP, so that's why this hormone would activate, So that's why there's a little bit of difference between all of them, just because the body needs to be able to differentiate like what type of response is needed, basically. But it all starts with the hormone binding the receptor, right? So, cool.
So then, looking at this question,
Oh yeah.
so hormone binds to receptor activity. Kinase enzyme, what would result? So phosphorylation occurs on the receptor and associated proteins. Would it occur on the receptor or like in the cell?
Well, it depends on like with the first one I drew, it's on the receptor.
Okay.
Yeah. Let's see.
Dimerization? I don't know what that means.
Dimerization. Yeah. Let's see.
I'm guessing it's not that one. 'Cause
Well,
maybe not.
yeah, so some of these, it's kind of funny. It actually could be that one, but, because a lot of these proteins don't look like this, they actually look like two subunits, like this. And when something binds, it causes them to come together and form a dimer, and that's how they become phosphorylated, is they basically like phosphorylate each other.
Okay.
But I think that's kind of beyond the scope of this class, but I didn't really see much of that, because I mean, in your book, or in the book, it doesn't even show it, and it just shows it like a simplified version. So, but I'm trying to remember, dimerization might be, I'm pretty sure it's the coming together. So let's just keep that one open maybe. Okay, alpha subunit binds to GTP, separates from beta gamma subunits. So,
It's part of it, right?
yeah, well, that's where you need, like the question, hormone binds to receptor, the receptor may act like a kinase enzyme. So, this is the kinase enzyme, but, so in a G protein, the receptor isn't acting like the kinase enzyme, it's activating a different structure, which is a kinase enzyme. But with this one, it is acting as a kinase enzyme. Does that make sense? So that's kind of where. So it's talking about a non-G protein one. So we could probably rule out C, right? And then, yeah, we'd rule that one out too. Because this is basically, this one cuts out this whole process right here.
Gotcha.
Right, and so anything involved with here isn't happening here. It's just phosphorylation activated protein kinase, right? So
it would probably be A, phosphorylation occurs in the receptor and associated proteins.
yeah, I think so. Because dimerization of receptor, which occurs with the chaser receptor, that might be true, but the question is what result occurs within the cell. And so not necessarily at the protein, because it says phosphorylation occurs at the receptor and associated proteins in the cell, right? So that would be my best guess, but let's see what it is.
Which is A.
Cool. So yeah, because B is technically true, I'm pretty sure, but oh, okay, yeah. No, because this is called tyrosine kinase. Tyrosine kinase. I was looking for that. That's the dimer one, and it may or not be involved with hormones. I don't think it is. Maybe it is. It's been a really long time, but it phosphorylates and then phosphorylates thing within the cell. But I don't know if it's a hormonal one. That might have been like why that one's wrong. But yeah, so. But I mean, I don't think most people in your class have heard of that, so I think it would probably make me more confused than anyways. But